Okay, good morning, students. Am I audible?
Yes, ma'am.
Yes, ma'am.
Fine. Okay. So all of you have joined, and we can start with the practical class. And before we start a new practical today basically we are going to discuss about the polymerase chain reaction. There's one thing that we have a like diagnosis of viruses like plant and human and animal viruses, both in theory also and a practical also. So in practical, I'll going to discuss the overview of a PCR and how practically we do it. And simultaneously in the next lecture in theory also, I'll start the diagnosis where in detail I'll discuss about the PCR and the types of the PCR and what are the different principles behind the PCR or different kind of a PCR. What is there are how many types of PCR are there and how we use the different kinds of a PCR for different purposes, right? So usually diagnostic techniques are similar in plant and human viruses, but there are few differences. So I'm going to take like one lecture for a diagnosis of a plant viruses and another lecture for diagnosis of a human or animal viruses in the theory. But practically, when we do like I'm talking, we are going for the DNA isolation. Where last step we did the DNA isolation. So, and the next step is basically the polymerase chain reaction. That how through PCR we identify a particular virus in a given sample. So that we are going to discuss today, right? So okay. So last time we discussed about the nucleic acid isolation or isolating DNA from the plant. So who is going to tell me that what are the problems that we face while isolating a nucleic acid from the plant? Yes, uh, Devanshi. Okay, Manaswani. Okay, Manaswani, you go on.
Ma'am, so one issue that might happen when extracting the DNA of plants is that, let's say, the whatever buffers or reagents that we use, if it's in not the correct pH, then that can
Mm-hmm.
destroy. It can affect the DNA a lot, and
Mm-hmm.
sometimes some mechanical error, there can be disruption of the molecule,
Mm-hmm.
and yeah.
Okay, yes, okay, fine, that's fine, that's right. What are the other things? What are the some compounds in the other students? Like, what are the some things, few compounds in the plant that hinder in the DNA extraction? Can you just name those compounds due to which the isolation of a DNA is not very easy, and sometimes we need to add certain additives to bind to those compounds so that we can isolate our DNA properly. Yes, Janvi. Janvi, Kshitij. Mhm. No one. So you guys are not studying that much, right? So okay, next week I'll have one test, and that test will include your both practical and theory part completed till now. So next, most probably we'll take test on next Thursday only, since we have a practical class, and that will give us enough time. So prepare well. It could be one word, one liner, 5 marks, 10 marks, anything. So you'll prepare thoroughly. There's nothing like that. There will be short answers only, or there will be detailed answer. It could be just one word answer also. So prepare thoroughly for your exam, and the things that we have done till now, both in theory and practical, they will come in the exam. So prepare well for your exam, and this will be the part of your assessment also. So next week we are going to discuss, or we are going to go for one assessment, right? Fine. So last time we did the DNA isolation, and we talked about that there are certain polysaccharides and polyphenols which are some compounds which are present in the plant species, and they cause some kind of a problems when we are isolating DNA. So also the DNA isolation from, or RNA isolation from the plant cells is difficult as compared to the animal cell, or as compared to the bacterial cells. So because there is difference in the structure of the plant cell and the cell wall of a plant, they are very rigid. So we need to go for a either the mechanical extraction or using some kind of enzymes or some kind of a chemical. Then the buffers, we need to be very careful. The buffer, CTAB buffer, is the main that we use for the DNA extraction in the plant, and we should be careful. We need to set a proper pH. Then certain additives such as polyvinyl, pyrrolidine, and other compounds are being added in the buffer also. And CTAB itself is a cationic detergent, and it is very commonly used in the DNA extraction. It is soluble both in water and alcohol, right? And its properties, it can be corrosive and toxic, so we should be very careful while handling this. We need to use proper precautions. Then we use a liquid nitrogen also in the DNA extraction. For it is used for crushing the plant material basically. So, and for removing phenolics from the plant, polyvinyl pyrrolidine is commonly being used in certain concentration. So we go for that. And the methodology is okay. I'm sure you all must have submitted your practicals also. I'll check after this class. And these are agrogel electrosources we perform to check the if integrity of our DNA, whether the DNA that we have isolated or RNA is in proper shape. And for the concentration also, we do the spectrophotometer. We use the spectrophotometer also to check the concentration of purification of the DNA. Then I also told you that beside using the classical method or a CTAB method for the extraction of a DNA or RNA, there are certain kits which are available in the market, and there are basically silica gel based isolation. They have certain columns, and we just need to perform the steps. It's very easy, and the time taken is also very less, and the quality of the DNA and RNA that we get through these kind of a kits is definitely in a very purified form. So when we are using DNA or RNA for molecular purposes or research purposes, then we definitely prefer to go for the kits. And it also depends upon the whether we have any kind of a funding available with us in the lab. So that was the DNA isolation protocols or RNA. Then we need to be more careful when we isolate RNA, because RNA degrades very quickly. So usually cold rooms are recommended, or it's just that we do all kind of isolation, and we place sample on the ice most of the time. We don't bring it to the room temperature, and as soon as we isolate it, we just store it at -80 degree or -20 degree for further use, right? So that was the isolation part. Now next part, so we are going for a stepwise. Like first we studied the symptoms, right? On the basis of symptoms, we brought samples from the field, and then we after that we did the mechanical inoculation, that we tried to maintain the virus on the host plant. Now we wanted to know whether that virus has been transmitted to the host plant or not. We need to perform some diagnosis. Then we did the RNA or a DNA isolation protocol, and then after the DNA has been isolated, right? It's a genomic DNA we isolate. It's not that we isolate the virus DNA. That is not possible. It's just when we isolate the genomic DNA of the plant, along with that the virus DNA also get isolated. Now we want just by isolating DNA, we cannot be sure, and we cannot know that our virus is there or some kind of a pathogen is there. So we need to go for a further detection methods, and there are either when we have isolated DNA, so definitely we go for a polymerase chain reaction basically. So there are certain guidelines and certain common steps to use the PCR. There are basically PCR and the RT-PCR. So reverse transcription polymerase chain reaction is being reverse transcription. One extra step is being performed when RNA viruses are being we are working on RNA viruses. And if we have a DNA viruses, we have isolated DNA, we can directly go for a polymerase chain reaction, right? So a polymerase chain reaction is basically very commonly, and it was developed by, invented by in 1984 by biochemist Gary Millis, and after that, its tremendous use in diagnostics and many other research has been done. I mean, it is very widely used technique for the identification of the pathogens or amplification of the other genes or other materials. So it is very commonly done, right? So this is this by the polymerase chain reaction. So it means it's kind of a chain reaction which is used to rapidly make number of copies of a specific DNA sample, right? So it's just the small amount of a DNA that we use in the PCR, and then by using certain primers for desired genes for viruses that we want to amplify, number of copies can be made, and it can be visible on the agarose gel electrophoresis that whether we have got the right amplification or not. So a a basic setup requires a lot of components are required. So usually we get all these components in the form of a kit from the there are a lot of industries, a lot of companies, they develop the PCR kits and a PCR enzyme. So enzymes we get from there, then the buffers also they come with the enzyme. Then the primers we design ourselves, or they are already available in the database, and we can just go and see, okay, this primer will work for our study, so that can be done, right? So basically what we the major part is the DNA template that contains the DNA target regions to amplify. That means the template which we want to amplify, right? So that DNA is required. Then we need a DNA, require a DNA polymerase. So it is an enzyme that polymerizes the new DNA strand. So basically this is a heat resistant Taq polymerase. It is very commonly used, and it can remain intact during the high temperature DNA denaturation processes also, which goes around 94 degrees Celsius, right? Then we need two DNA primers also, which are complementary to the 3' end of the each of the sense and antisense strands of the DNA. So that's why we have a forward primer and we have a reverse primer. So we have a two primers, right? Then okay, then primers, I told you that I'll definitely take one lecture on primers for you. It is not very difficult, but yes, there are certain requirements when we try to develop, or when we try to design primers, that whether we are designing specific primers or we are looking forward for a degenerate or a group specific primers that can amplify the major viruses in a particular group, right? So it totally depends upon the kind of work that we are doing. Then okay, we need a building blocks, basically DNTPs. Those are called DNTPs. With the help of a DNA polymerase, they synthesize the new DNA strand, and they are like DATP and DCTP, DGTP and DTTP. So in the form of a DNTPs in 40 millimolar and 10 millimolar solution, they are being provided. They come as a 100 millimolar stocks, and we can then change the stock as per our requirement, right? Then a proper solution which provide a suitable environment for the stability of the DNA polymerase. So a proper buffer is being provided with the kit. Then magnesium ions, basically in the form of magnesium chloride. These are the these ions, they bind to the catalytic site of the enzyme and then increase its power to perform the reaction. So they work increase the efficiency of the Taq polymerase that we use. So these ions are in some solution. They also come in the form of solution, and we use them. And then Taq polymerase by using DNTPs on growing DNA strand after the annealing to the primers at a particular temperature is being done. A proper reaction takes place. So it is an exponential process, basically, like from two strand they make a four, then from four they make a eight, from eight they make a they exponentially they increase. So a reaction is carried out depending upon 10 to 25 to 50 microliter, whatever is our requirement. If just for the checking purposes in the beginning we are doing, because we don't know whether the amplification will come or not, right? We are not yet sure. So we'll start with the minimum volume, maybe 25 microliter. This is just for checking whether virus is there. So if the amplification comes, we can again go for a repetition of the PCR, so that we have a larger amount of a DNA for the elution purpose, right? So most thermal cyclers, they have a heated lids to prevent the condensation at the top of the reaction tubes. And yes, older thermal cyclers, they were not having these kind of a heated, or they were not hot start PCR basically. So sometimes the layer of oil on the top of reaction mixture was added, so that our reaction mixture does not vaporizes, right? So reaction mixture is goes like this. We have our DNA template. So usually 5 microliter, or it totally depends the concentration which we need. If our DNA is in a good concentration, we can take a smaller amount also. Then DNTPs, it is also totally standardized with a particular reaction, 1 to 2 microliter. Then primer 1 and 2, 1 microliter each. Magnesium chloride also 1 to 1.5. Tag polymerase, and water to make up total volume. So these are the different components in a particular PCR that we use. And there is one other important thing that usually we make a master mix. When we run a PCR, there is when we go for a one or two reaction, we can put the things individually. But when we are going for PCR for more than three reactions, then we prepare a master mixture, so that because बार बार आप tip use करोगे और enzyme use करोगे, तो that will lose your enzymes also, because the enzyme is very costly and we are using in a very small amount. So we don't do that. In order to avoid that, we prepare a master mix, and in the end, we distribute that master mix in the different tubes, and we add our DNA template, we mix it properly, and then we go for the PCR. So master mix preparation is being recommended, right? So typically PCR consists of it depend a number of cycles. Like if our the desired amplification is around somewhere 500 base pair to 1000 base pair, so 30 to 35 cycles or even 28 to 30 cycles are enough, right? Sometimes. So it totally depend upon the kind of a fragment we are using, kind of a amplification we are desiring, right? So the cycling is often preceded by a single temperature step at a very high temperature, 90 degrees Celsius, and followed by one hold at the end of the final product extension or a brief storage. So there's a cycling there's a machine called thermal cycler in which we set the different temperature, first for a denaturation and then annealing, and then in the last days there's an extension also, right? So different temperatures are being set, and the number of times the cycles we want, depending upon the product that we want, size of the product or the concentration, number of the cycles can be set accordingly, right? So initialization. So this step is required for a DNA polymerase that require heat activation by the hot start PCR. So basically it consists of a heating the reaction chamber to the 94 to 96 degrees Celsius or 98 degrees Celsius. If extremely thermostable polymerases are used, which is then held for 1 to 10 minutes. So it is not required in all the cases. Then we have a denaturation step. So this is usually the first step in the thermal cycling event, and it consists of a temperature 94 to 98 degrees Celsius for around 30 seconds, up to 30 seconds. So this step causes DNA melting or a DNA denaturation, and our double stranded DNA template is it break it by breaking the hydrogen bonds between the complementary bases, and we get the two single stranded DNA molecules after this first step. Then next step is annealing, and annealing is very important. And annealing temperature, basically annealing means our primers will bind to the template, DNA template at this time. So when we design primers, we make sure the annealing temperature, like we calculate the melting temperature of the primers first, right? Which should be ideally somewhere between 55 to 65 or little more, but ideal temperature should be there, right? So we need to be very careful when we prepare primers that GC content should not be very high, because more in GC contents will increase the melting temperature. And if we get a particular melting temperature for a primer when we calculate, so annealing temperature is usually 2 to 3 degree lower than the melting temperature of a primer, right? So this annealing temperature, many times we need to standardize for a different viruses that we are using, right? Then according to the different primers that we are using. So annealing temperature is very important. So it should not be very high जिससे बाइंडिंग ही ना हो, और it should not be very low जिससे नॉन स्पेसिफिक बाइंडिंग भी हो जाए. So we need to be very careful when we design a primer that annealing temperature should be an average annealing temperature. It should not be very high or it should not be very low, right? So the reaction temperature in the annealing temperature is low to around 50 to 65 degrees Celsius. So at this temperature, it allows annealing of the primers to each single stranded DNA molecule templates that we got after the denaturation process, right? Then two different primers are, we include primers in the reaction mixture, right? Then how we design primers, that I'll tell, because it is itself is a whole one lecture. You need to take care of a lot of things while you design primer. So maybe next time I'll take the designing of a primer also, right? Then this is the TM. The melting temperature is calculated by this formula that so it is critical to determine the proper temperature for the annealing step, because efficiency and specificity, they both are affected by the annealing temperature. So this temperature, as I told you, must be low enough to allow for hybridization of the primer to the strand, but then high enough for the hybridization to be specific, because बहुत low होगा तो non specific binding भी होगी. That means the primer should bind only to a perfectly complementary part of the stand and nowhere else. So if what happens if the temperature is very low, primer may bind in imperfectly, or if it is too high, it will not bind at all. So a typical annealing temperature is around 3 to 5 degrees Celsius below the TM of the primer that we use. So stable hydrogen bonds between the complementary bases are formed only when the primer sequences they closely matches the template sequence. So during this step, the polymerase bind to the primer template hybrid and it starts the DNA formation. Then the next step is the extension or elongation. So temperature at this step again depend upon the DNA polymerase that we are using in the reaction. So mostly we use a TAC DNA polymerase and they are very thermostable, and they're 75 to 80 degree तक उनकी efficiency अच्छी रहती है. Though the recommended 72 degrees commonly used with that time, but usually they are good at that time. So at this step, the DNA polymerase they synthesize the new DNA strand which is complementary to the DNA template by adding the DNTPs that we have used in the reaction mixture, right? So they use those DNTPs, and then this is the final elongation. This is optional, not for every PCR recommended, but we go for this. So this is performed at temperature 70 to 74 degrees Celsius. So at the end, at the last minute, I mean, there to ensure any remaining single stranded DNA is fully elongated, just to give a chance. अगर नहीं हुआ है तो इस step पे वो elongation भी आपकी हो जाए. So this is for that step. So now to check whether the PCR successfully generated the anticipated DNA target region, we do the agarose cell electrophoresis. So we know, for example, I'm looking for the I'm looking for the prunus necrotic ring spot virus, which is affecting the stone fruits, plum, peach, and apricot. Now I know the code. I've designed the primer for the code protein gene of this virus, and I know the code protein gene of this virus is 800 base pair. So if I'm getting amplification around 800 base pair, or maybe somewhere 800 to 850 base pair also, then it will give me indication that the code protein of the virus has been amplified and the virus is present. If I don't get such amplification, I get maybe 200 or I get maybe even more than the 1000. So that means there is something wrong. Maybe the binding is not proper. I might check the annealing temperature. I might go for a gradient PCR where number of annealing temperatures I can check in a one reaction. So I can do those things to check whether the sample is positive or not, right? So this is the gel electrophoresis at the end, that if you run a ladder, like in M side, you can see the marker or a letter that we call, and it's labeling, and the lane 2, 3, 4 are your samples that you have run in the PCR. So this is somewhere 321 base pair or 320, 300 base pair, more than 300. So it could be 300 and between 300 and 350 base pair. So if this amplification is desirable or it is as per our expectation, we can go for the further processes of DNA purification. DNA, we need to elute this DNA from the gel, right? We need to take out this DNA from the gel, and then we'll go for a cloning, and then in the end we'll sequence the sample and we'll see whether the virus is there or not. So this is a long process. It's not that only after the PCR you'll be able to know that okay, this virus is present, sure shot. Even if you get sometime the desired amplification, there are chances, there are 10 to 15% chances that at the end when you will do sequencing, there's something else, maybe some plant gene product or some other plant gene which is being amplified falsely, right? So इस तरह से false positives बहुत बार होते हैं, and only at the end of the sequencing you come to know that this is not which you were looking for. Then you can repeat the process and you'll check by some using some other annealing temperature, or maybe there are sometimes other specific primers that you can use. So these kind of problems can happen if you're using degenerate primers. That means primers which can amplify a broader viruses or number of viruses in one particular genera or in one particular group. So there these chance. But if you're going for a specific viruses for a particular code protein gene, then these kind of false positives are very less. So it usually does not happen, right? So optimization, as I told you, we need to optimize PCR for every kind of a reaction, right? Okay, for one virus, for example, I'm working on Pune cytotic ring spot virus, so I need to optimize the PCR for this virus. If I'm working on apple mosaic virus, so again the optimization will be required for the PCR for that particular virus, because we are working on two different, because the annealing temperature could be different for both the primers that we have designed. जरूरी नहीं है कि सबका annealing temperature same हो. They could be they could be the another problem also, that the non-specific applications more at the lower annealing temperature, so we might need to change the annealing temperatures to little higher side also, so that they can bind to the primers properly, right? So in practice, PCR can fail for a number of reasons. Yes, due to the sensitivity. This is a very sensitive process, and any kind of a contamination can interfere with the amplification process. So this is very important, that when we do such kind of a high and sensitive, we use such kind of sensitive techniques or sensitive equipment, so we should be very careful. The handling should be very careful. There should be gloves all the time. There should be it is recommended to prepare master mix in the cold rooms or at the lower temperature, so that the degradation of DNA or RNA is not there, right? So the PCR components we need are DNA samples, we need primers, and we need a nucleotides, which is provided in the form of a DNTPs, then a PCR tube, which are being fitted in the PCR block, thermal cycler, buffer, and the tag polymerase, right? So first step, as I told you, is the separation of a strand, which means denaturation. Second step is the primer, they bind to the template, which is called annealing. And the third step is the extension, synthesize the new strand and extension of that particular DNA further. So this is how the PCR works. And okay, uh, this is again the same things, like for new sample, new virus, and new gene, we need to standardize the PCR. We need to look for optimum annealing temperature. We need to sometimes standardize the concentration of the primers. Like for some, we need a lower concentration of the primers, somewhere we need the higher concentration, even the concentration of enzymes also sometimes, right? Then I told you the DNTP, they are being added. Their function is to expand the growing DNA strand with the tag polymerase, MgCl2 enzyme cofactor, tag polymerase, and gradient PCR. So what is gradient PCR basically? So in gradient PCR, there are different rows in the PCR, right? There's a block in the PCR. You can fix your PCR tubes and you can run. So when you want to check different annealing temperatures in one reaction, because otherwise what happens, you will run annealing temperature, one PCR reaction for one annealing temperature, maybe annealing temperature we are expecting somewhere 55 to 57 degrees Celsius, right? So first reaction we are running at 56 degrees Celsius, we don't get any amplification. Next we want to check 56 or 57 also, or in between also. So we need to go for a 3 or a 4 PCR reactions. But in a gradient PCR, they are designed in such a way that if you put sample, you just need to set the gradient, that you need to check the temperature from 55 to 57 or 58 degrees Celsius. So accordingly, in separate rows, we'll put those PCR tubes. So in one reaction, it can check the different annealing temperatures, and we'll get a result. Like this is basically useful when we are standardizing the annealing temperature or standardizing the PCR for any annealing temperature that we are using. So वहाँ पे क्या होता है, एक ही step में we can check different annealing temperature, and जहाँ पे हमें लग रहा है amplification आ रही है, when we run the gel, we can go for the more we can use that annealing temperature for amplification for the gene put that we do, right? Then okay, one approach again, then as I told you, we need to, one approach assay optimization is to determine the optimal annealing temperature of the primer by testing identical reactions containing fixed primer conditions, so that can across a range of annealing temperatures. As I told you, this can be done. So if we have a gradient block available with us, so in this we can do in one reaction only. So procedure is same. We need a DNA or RNA we need to extract. Then if we are going for RNA viruses, we need to do reverse transcription, which is one step by using reverse transcriptase, and then we'll go for a PCR. by preparing our master mix and going for a thermal cycling condition. We run the sample in the block, and it takes around 2 to 2.5 hours depending upon the number of the cycles that we are using, right? So after that we can check the result by agarose gel electrophoresis, and whatever amplification come, it is desired or not, this gives us indication that this virus is present or not, right? So basically, what we need for the polymerase chain reaction to do identification of the virus, we need two primers which are either specific for that virus, or we need any degenerate primer which are specific for the that particular genre of that virus. So that will amplify all kind of viruses which are under that particular genre or genus, right? So primers are very important, we should have. So and primers are usually, see, like we have a sequences, when people they identify new sequences, they submit it, there's the NCBI, they have a nucleotide sequence database there, so people they submit their sequences there, right? So we can get the primer sequences from those open banks also, they are available, we can use them. If we get any amplification or we just need to refer, or we cite those references in the paper if we are writing some paper, or we can design our own sequences. That too, we need those sequences from the database. We analyze those sequences, and we generate or we develop our own primer, and its synthesis is being done by the companies. They do this on a regular basis, so we can give our primer for the synthesis. Once we have designed, we can give for the synthesis to any company. They'll synthesize it and we'll get a primer and can be used, right? So to run a PCR primers, we need a primers, we need enzyme, we need a buffer, we need a DNA template, right? So all those things are very important to do a PCR in the lab, basically, right? So this is all for the PCR, that how PCR is being done. I have one video basically, so let's see if it explains further or not, right? I'll share my screen. Is it audible or not? Is it audible or not?
Ma'am, we can still see the slide.
Yes, ma'am.
But
The slide is visible.
Yes, ma'am.
just a moment, I shared my screen. Just a moment.
Ma'am, I think you'll have to. Yeah, never mind.
Yeah, but tell me about the audible. Is it audible or not? Is it audible?
No, ma'am.
No,
No,
ma'am.
just a moment. I might have to do one step further. Okay. Now, is it audible now or not?
No, ma'am.
No,
It's not.
ma'am.
No, ma'am.
Just a moment.
Forensics,
Now,
DNA sequencing, and diagnosis.
Yes,
of genetics.
ma'am.
is it audible now?
Before starting
Yes,
PCR,
ma'am.
I'll need to design primers to target the region within the human genome
Is it audible now?
Yes, ma'am.
that I want to amplify. Primers are short sequences of DNA that bind to specific regions within the genome and tell DNA polymerase where to initiate replication. So here I have a fragment of the human genome, and in the center is the region that I want to amplify. In order to amplify this region, I'll need to design both a forward primer and a reverse primer. My forward primer will anneal here, and DNA polymerase will amplify in the 3' direction. Then I'll design a reverse primer that anneals here. And again, DNA polymerase will amplify in the 3' direction. After many cycles, predominant resolution like this. The sequence will exactly match my amplified region. And the sequence here will be identical to my forward primer. And the sequence here will be identical to my reverse primer. So now it's time to set up the PCR. I'll be adding the components from largest volume to smallest volume. First is water. I'll be adding these components to the PCR tube, which is very small and has thin plastic lining to enable it to change temperature quickly. The reactions also happen in small volumes so that they can change temperature very quickly. Next, I'll be adding the PCR buffer. This buffer contains tris HCl and KCl, and it's meant to stabilize the pH between 8.3 and 9. The buffer is at a 10x concentration. Its final volume within the reaction should be 1x. So I'm adding 2 microliters to my 20 microliter reaction. Next, I'll be adding the DNTPs. These are the building blocks of DNA, and this is what DNA polymerase is going to be synthesizing together as it creates the new strands. Next, I'll add magnesium chloride. This is important for the reaction. It's an important stabilizer for the DNA backbone and stabilizes its interaction with DNA polymerase. Next, I'll be adding my DNA. Just going to add 1 nanogram of pentanoate. Next, I'll need to add my primers. I'll be adding the forward primer and the reverse primer separately. The final concentrations of these primers will be 1 micromolar, but you may need to troubleshoot this for a different PCR. You may need more or less. The first is the forward primer. And next, the reverse primer. And finally, I'll add DNA polymerase, which will carry out the DNA synthesis reaction. There we go. And once I've mixed and spun down my sample, I'll be ready to go. I just finished showing you all of the components that go into one PCR reaction. Oftentimes, all of the components of PCR, except for the template and primers that you bring in, are pre-assembled into what's called a master mix. Master mixes make pipetting more accurate and convenient and should be used when setting up multiple PCR reactions. I used a master mix to set up these 3 PCR reactions. Before I set up the PCR program, I want to quickly review the 3 steps of a PCR cycle. In the first step, double-stranded genomic DNA is up and separated into 2 single strands. This step is called denaturation, and it occurs at 95 degrees Celsius. Once we have 2 single strands, we lower the temperature to the primers we designed to bind to the complementary regions within the genome. This step is called annealing. And in RPCR, this is happening at 68 degrees Celsius, but this may differ based on the melting temperatures of the primers you design. Then, in the last step, DNA polymerase moves in and begins replication, generating 2 pieces of DNA. This step is called extension, and it happens at 72 degrees Celsius. Now it's time to set up the program. Next, I'll need to write a PCR program into the thermocycler before adding my sample. Here's the thermocycler screen that shows the schematic of the PCR reaction, with each step from left to right, and steps 2 through 4 here will be repeated within each PCR cycle. So the first step is the initial denaturation step. I want this to be at 95 Celsius, and I want this for 5 minutes. Next is the denaturation step within each cycle. I want this at 95 Celsius again for 30 seconds. Next is the annealing step. I'll calculate the temperature for this using the average of the TMs of each of my primers. So this will be at 68 Celsius for this primer pair, again for 30 seconds. And then the last step of the cycle is extension. I want this at 72 Celsius for 30 seconds. Then this cycle will repeat 34 times. When the cycle is over, I'll do a final extension step so that DNA polymerase has an opportunity to finish up any copies that weren't completed. So I want this at 72 Celsius for 7 minutes. And then finally, I want the thermocycle machine sample, so they stay nice and cool. Now I'm ready to add my sample to the thermocycler. I'll place them here in the center. I'll close the lid and tighten it. You may not need to tighten the lid on your PCR machine. And I'll click run. I'll come back when my PCR samples are ready. My PCR program is now complete. I expect that my target DNA sequence got amplified many times. One of the most common ways of analyzing your PCR is to run a DNA cell. You've now seen me assemble and run a polymerase chain reaction. I hope this helps you in your laboratory work. Thank you for watching.
Hello. Am I audible?
Yes, ma'am.
Ma'am. So this was how we do it in the lab, and we have also a biored PCR here. One of the PCR is a biored, which is the oldest one that we use. So this is not a complicated one. I guess once you learn how to do it, anyone can do it. You just need to follow the steps and the precautions. And yes, sometimes we need to standardize PCR many times because we don't get a desired application, and sometimes we might need to redesign a primer and to look for a different primers because sometimes different kind of a problems also arises. While we design the primers, right? So they are not working, so we need to look for a different primer. So this is how a PCR is being done, and it is one of the routine tests that we do for the diagnosis of the plant viruses. Yes, when I'll discuss the diagnosis of a plant viruses, I'll discuss the ELISA also because that is the most common one when we have a large number of the samples. Because PCR is not very economic. Yes, tech polymerase and all the components, they are costly, whereas ELISA or a serological testing is much cheaper as compared to the polymerase chain reaction or a molecular testing. But in some cases, we might require only molecular testing, so we go for that. But otherwise, testing by ELISA is also one of the best way to check the virus, whether it is present in the sample or not. Right. So this is how we go for it. And one of the routine practical that we do, or routine experiment that we do in the research labs or in the teaching labs also, we tell students that how they do PCR step by step. We just give them a sample. It basically how it starts like we ask them to isolate a DNA and then use that DNA. We give them primers to check a particular virus in a sample. So this is a step-wise procedure that from step 1 to step and then we ask from them to for a cloning also, that okay, you clone this in a suitable vector now, and prepare your competent cells also, and do your cloning experiment also, and then let's see what it comes out after the sequencing is being done. So sequencing, we don't have a sequencer in the labs here, so for that we outsource it. There are many industries which do these kind of a work for a very less price. I mean, these days, I guess for a 500 base pair, we just need to spend 500 bucks only. So it's not very costly, and we very commonly do this kind of a experiment to check the viruses in the sample, right? So this was all about today's lecture, the practical lecture. I was checking that last week's practical, not all of you have submitted. I don't see the files, I guess. Adhish and Athoiba, I don't see your file. Rest of Namrita also, you have viewed, you have not submitted. And Tridev turned late. Okay, you submitted. Let me check. Yeah, Tridev has submitted.
Yes, ma'am, I'm submitting. Right.
Yeah, please, others, please submit. So I'll check the files. And for this practical also, I just a moment. For today's practical, you'll write. Where is the practical I've given you? Just a moment. Okay, you'll write an experiment which will be titled as PCR for the identification of virus from the plant sample. So for this, I've just told you the step. You'll arrange those steps yourself. What will be the requirement? What will be the sample? If you want to select a RNA virus, then you'll select a RNA and you'll perform a reverse transcription one reaction, and then you'll go for a PCR. Or from a DNA virus, you can directly perform the PCR. So a proper methodology to be written for the polymerase chain reaction when you do for the identification of the virus from the plant sample, right? So this is for today's practical. You're going to submit these practicals maybe before the next practical class. You can submit. Also, there's one important announcement that I want. Next week you're giving exam, right? Be ready with the exam. And also, let's Friday, because we don't have a lot of syllabus in this course to be done. There are very few things. So I thought that Friday's class will keep for the discussion. We'll take one topic, and a topic could be directly or indirectly related to this subject. So we'll take one general topic and we'll have a group discussion on this topic. So tomorrow's topic I'll be uploading on the team. So come prepared with the topic, and we'll have an open discussion forum kind on the topic, like anything about a topic, whether it could be a pandemic, it could be some old epidemic. And I guess the recently I was checking the news that in Kerala, again, there is a Nipah virus case being found. One boy died from this virus. So, okay, so let's, for tomorrow's topic, let's take the Nipah virus outbreak in Kerala, which took place maybe 2, 3 years back. It was in 16 or 14, I don't remember. So, just go through the papers and just go through the material which is related to Kerala virus, Nipah virus outbreak, that why this virus outbreak was there, what they did, what Kerala government did to contain the virus, why the virus did not spread outside the stage, how they were so efficient in containing the virus, and then the point that why this virus is re-emerging again. I mean, why after so many 3, 4 years again the cases are being, and was there any kind of a one point that they narrow down to some point that what was the cause of that virus, even why that virus outbreak that started in that Kerala. So all those factors, try to go through. Even I'll go for some papers for this. I don't know if you all have watched or not. There's one movie on this Kerala virus outbreak, which explain all the sequential facts and how they came to the index patient and how they located the other people who came contact with that patient, how they did that and how they were able to contain that virus. So it's a very good movie on the corona. Similarly, there's a movie on the Ebola virus outbreak also. So if you want to, you can watch that. But there are a lot of papers also available on the Nipah virus outbreak in the Kerala. So tomorrow we'll have an open discussion on the Nipah virus outbreak in Kerala. Right now, for your attendance, please all of you switch on your cameras. Adhish Mishra,
Present, ma'am.
Athoiba,
Present, ma'am.
excuse me, Ayushi,
Present, ma'am.
Sam,
Present, ma'am.
सैम के बाद कौन है देवांशी
Present, ma'am.
आपको आज तो किसी ने बाहर नहीं निकाला देवांशी
Sorry, ma'am.
yes last week you were saying that students are I mean they are just removing you from the team तो अब आज तो नहीं किसने निकाला बताना मुझे कौन निकालता है तुम्हें ठीक है
Okay, ma'am.
इशिता अरोड़ा जानवी सिंह क्षितिज
Present, ma'am.
Manaswani, yes, I can see you both. Then we have Namrita Paul. Namrita is there. Nandini is there. And we have after Nandini, we have Nehal, Rupal,
Present, ma'am.
Sahiba, Sanjana.
Present, ma'am.
Present, ma'am. Rupal, Sahiba,
Present, ma'am.
and Shakti, yes,
Present.
Shakti is there. Sanjana is there. Shivani,
Present, ma'am.
Tanvi, Tridev is there.
Present, ma'am.
Yukta and Yuvraj and Vivek.
Present, ma'am.
Fine. All of you are there. So, okay, today all students are present. Fine. So